everyone, welcome to science class. Today we're going to be learning about the structure of DNA. Now we've been learning about cells for the past few weeks, and we know that cells contain DNA. So what you're going to need for this experiment is rubbing alcohol. Your parents might have to help you get this, but I'm sure they'll be willing. A cup of some sort, scissors, a pencil, 3 types of pens. It doesn't matter as long as they're different colors. I'm using a blue pen, a black pen, and a highlighter. A lid, and some type of straight edge. And you're also going to need, I forgot to grab it, a coffee filter. Okay, so we're going to take our coffee filter and straighten it out. This coffee filter represents chromatography paper, which is what they would use in a real investigation. So this is our chromatography paper. We're going to cut a square out of our chromatography paper. You want this square to kind of be larger because we're going to have to manipulate it later. So we're cutting our square. Doesn't have to be perfect. So we have our square. Now we are going to take our pencil. It's important that you use only pencil for this part of the experiment. You can take your pencil, take your straight edge, estimate about 1/2 inch up, draw a line, so we have our line. Now we are going to take each of the pens and draw a dot with it. You want this dot to be kind of bigger. You want it colored in. So there's our first dot. And you want them, you're going to want the dots to be pretty spaced out because we're going to end up putting it in alcohol. So we're not going to want our dots to rub together because it'll make the experiment not accurate. So we have our 3 dots. Now we are going to, couldn't find my paper clip. We're going to make, wrap it around, paper clip it together. And then we're just going to set that down. We're going to guesstimate how much alcohol to put in. You don't want the alcohol to go over where your dots are. Then it'll cause it to run into the alcohol. So we're going to pour our alcohol in. That's good. Then we're going to place our chromatography paper in here. Now, this type of experiment is often used by forensic scientists, and they use it at like crime scenes. If a letter was written or a pen was in the person's pocket, they can collect the DNA off of it, and the pen will almost like separate, the different colors separate, and it causes the particles to match the particles that are at the crime scene, and their DNA is often embedded in parts of the pen, and so they match the chromatography paper to the crime scene, the DNA that was there, and that's how they often get a match. It's often used during murders or criminals fingerprinting. Chromatography paper can be used for that. So you're just going to let it process. This is going to take about 20 to 30 minutes. And then you're going to take it out. Just take it out. Wipe it off. Of course, we didn't wait 20 to 30 minutes. But I have done a previous one. And the final product will look like this. You can see how the black pen was made out of navy blue and black, and navy blue being the primary color, the blue pen was made out of a lighter blue and a dark blue. And surprisingly, the highlighter isn't made out of anything except for pink. So we can see the pink is very, very light because it didn't have very many particles to separate. So the more particles it has, the darker it's going to be. But this is chromatography paper again. We've learned about DNA, what it's used for in real life. So now you can go ahead and take your quiz.